Hello, this is our lecture number 8. We're going to talk about a very interesting story in the history of psychology. It's the story of a man by the name of Phineas Gage who literally changed the face of psychology and the study of behavior. And that will be as an introduction into a little bit about brain anatomy, and we're also going to get into how cells communicate with one another, something that we call the action potential, and how the action potential is so important in terms of understanding both very simple behavioral responses as well as more complex behavioral responses. So let's begin this analysis. This is Phineas Gage that you see right here, the man who changed the face of psychology. And what you see there is Phineas Gage holding something that's called a tamping iron. This is an iron rod that's approximately, well, it's about 1.5 inches, almost 2 inches in diameter. I think probably about 4 or 5 feet long. And one thing you should notice in this picture right here is that Phineas Gage, his eye, his right eye, is gone. And this is a consequence of an accident that occurred a number of years ago that I want to tell you about, and that you have by now should have taken a look at a videotape that I have on this very interesting historical accident. So this story of Phineas Gage is an incredibly important one. 1848, Cavendish, Vermont. Phineas Gage is the foreman of a crew of individuals that's working on this railroad line that's being built between Boston, Massachusetts, and ultimately going up to Montreal, Canada. So it's going through the state of Vermont. And he's the foreman, he's a dynamite specialist, and he's the leader of this group, very industrious, very hardworking individual. And there's an accident that occurs. That tamping iron that you saw him holding in that previous picture on the previous slide, that tamping iron is used in order to tamp that dynamite into that hole. It explodes. What happens is that tamping iron goes through his cheek and then through his brain and then out his head. And here's a reproduction of that here. This is about where that tamping iron went. And again, through the cheekbone, then ultimately in the back of the eye, severing all the nerves that go from the eye to the brain, and then ultimately out through the frontal area of the brain. So he lives, which is a medical miracle, but this very serious, very energetic, very industrious Phineas Gage now becomes very childlike, juvenile in terms of his behavior. He can't even carry out simple plans. His behavior becomes very whimsical and indeed very capricious. This is such an important story in the history of psychology because it really establishes the first documented evidence for there being a brain basis for our behavior, for psychological processes. This incident was written up both nationally and internationally in newspapers. Again, he lives for 12 years after this incident, manages to make a living by being involved in a traveling carnival where people would actually pay to see Phineas Gage, to see that he was alive, to see him holding that tamping iron, and to learn more about his story. The story of Phineas Gage, then, a very important story. Here's the actual site of the accident that you see right here in this gap. This is the granite that they were blowing up. This is Cavendish, Vermont, very small, beautiful village in the south central Vermont. And this is that you see right here. This was an actual cartoon that was in a local newspaper at the time. Take a look a little bit more about Phineas Gage. The actual accident itself is memorialized on this plaque on the village green, where you can see the important dates here, and a little write-up about this particular incident. This is the actual newspaper article that you see here, and just let me read this for you. It says, Phineas Gage, a foreman on the railroad in Cavendish, was yesterday engaged in tamping for a blast. The powder exploded, carrying an iron instrument through his head, 1 1/4 inches in circumference and 3 feet and 8 inches in length, which he was using at the time. The iron entered on the side of his face, shattering the upper jaw and passing back of the left eye and out of the top of the head. The most singular circumstance connected with this melancholy affair is that he was alive at 2 o'clock in the afternoon and in full possession of his reason and free from pain. This was from the Ludlow newspaper at that time. And again, this is something that attracted enormous attention throughout the world. This is the actual skull of Phineas Gage. It is in the Warren Anatomical Museum at Harvard University. You can actually see this. I had the opportunity to see it a few years ago. And again, here is the skull that you see right here. And here's the actual tamping iron that was used. So again, this is something that's been preserved, which you can see. So again, this provides the first indication that, if you manipulate, destroy the brain in some way, then it's going to result in changes in terms of behavior. And that really prompted psychologists to become much more interested in the study of the relationship between our brain and behavior. So let's learn a little bit here. Let's get some of the basics down. If you take a look at what happened in terms of Phineas Gage. Now, this is a view of the brain. This is the front of the brain that we see right here. This is the back of the brain. We're viewing this. This is the side view of the brain that you see right here. Okay, here's the top of the brain. Here's the bottom of the brain. This is called dorsal, this is called ventral, this is called anterior, this is called posterior. Here's the cerebral cortex of the brain that you see right here, the outer portion of the brain, the so-called smart part of the brain, as we will see. And this is inside, underneath the cortex of the brain, what is often referred to as the old brain, the thalamus, the hypothalamus that we see here, the hippocampus, the amygdala, the olfactory bulb. These are all considered to be parts of the old brain. So the tamping iron goes right up through here. And essentially what has happened in the case of Phineas Gage is the smart part of the brain, which normally inhibits the impulsive, emotional part of the brain. Those pathways that connect between this smart part of the brain and the more emotional part of the brain have been severed. So now there's nothing there to inhibit the emotional behaviors, these childlike behaviors that were exhibited by Phineas Gage. Ordinarily, that's what happens. The frontal area, the smart area, can inhibit what is going on in terms of these emotional responses. But now, without that inhibition, Phineas Gage becomes a very different individual, a very childish individual. So again, this is teaches us a little bit about the old brain down here and what we call the new brain. And by new, what I mean is in an evolutionary sense, this is a more recent evolutionary adaptation. So your brain, I think the best way to view it is in terms of a series of ice cream scoops. That's the analogy that I want you to think about, as we go along. You are really the top scoops. These are the most recent changes that have occurred in terms of our phylogenetic, our evolutionary history. Those other scoops that you see here Again, those have been there much longer. Through evolutionary time, now we have these really important cognitive functions that are involved with thinking and decision making that are added really as new scoops and placed on top. So here's old brain here, here's new brain right here. So again, try to think of your brain in terms of this particular analogy. I think that it will help you. So those lower brain areas that have been there for so very long period of time, involved in basic biological processing, breathing, rhythm, body temperature, homeostasis. These are things that really have been left unchanged. It's these recent adaptations here in the case of Homo sapiens, and we discussed a little bit of this when we were taking a look at Darwinian evolutionary theory. So that's how I really want you to try to conceptualize the brain. So the four primary lobes of the brain, let's make sure that we have a little bit of understanding of that. Again, here's our frontal lobe area, really the smart part of our brain. This is what's referred to as Broca's area right here. That's involved in speech formation. Here's our primary motor cortex that's in the blue that you see here. Again, our voluntary movements. Here in the green is what is called the somatic sensory cortex. Again, bodily sensations. Here's the parietal lobe, which has many different functions, as we're going to learn, but especially in terms of certain aspects of visual processing. In the back of the brain that we see here is what's called the occipital lobe. This is the primary spot for visual processing. The primary area where stimuli from our eyes are transferred. This area that you see right here is called the cerebellum, involved in basic motor control. Here's our spinal cord and our brain stem that you see right here. Here's the temporal lobe. And in the temporal lobe is what's called Wernicke's area right here. This is involved in the understanding of speech. Our primary auditory cortex is here. You will see as we go along that the temporal lobe very much involved in things like face recognition, for example. So these are the primary lobes in the sensory motor and visual cortex of the brain. So you should have some familiarity with these basic lobes of the brain. Here's another way of classifying different brain areas into what we call forebrain, midbrain, and hindbrain. All of this area that you see right here is what we call forebrain. The area that you see with this cross-hatching right here, this is called the hindbrain. And this area that you see right here with this this right here, this is what is referred to as the midbrain. Okay, so again, this is all forebrain. Okay, everything that's unmarked. This is all, you know, pons, medulla, cerebellum, right? This is all what we refer to as a hindbrain, and this is all midbrain that you see right here. So again, this is just another way of classifying these various important parts of the brain. So let's talk a little bit about the cells of the nervous system. Take a look at this diagram that you see right here. We have approximately 100 billion individual neurons. Okay, the brain is composed of these neurons that are broken down into, again, what we call, these cells are either called neurons or they're called glia. Glia are support cells. They don't really communicate over very long distances. They're principally involved in the nourishment of individual neurons and making sure that certain kinds of waste products are promptly removed from these neurons. So if you take a look then at this diagram, our cortex, cerebral cortex, is about 12 to 15 billion neurons. The cerebellum, about 70 billion neurons, and the spinal cord, about 1 billion neurons. So behavior really depends a great deal upon communication between these neurons. So we need to understand a little bit about the structure of these neurons, and again, how they go about mediating different kinds of behaviors, whether they are relatively simple kinds of reflexive responses, or whether they are more complex kinds of behavioral responses. We need to understand a little bit about these neurons in terms of how they function. So the neuron, we call it the building block of behavior, and this is probably information that you've seen a number of times before, but I want to make sure that everybody's on the same page. So let's make sure that we go through these things and understand these various parts of the neuron. This structure right here is referred to as the soma, or the cell body, and you can see the nucleus that's in the cell body. These are mitochondria that you see right here. These are what are called dendrites. This is the receiving part of the cell body. This is referred to as the axon. You see right here, the axon is covered by something called the myelin sheath, which helps to protect the axon, but is also very much involved in the conducting of neuronal impulses that go from this cell body down the axon and then ultimately to these axon terminals, or what's also called the terminal buttons. So again, the myelin sheath covers the axon. We have these little gaps that you see right here. These are called the nodes of Ranvier. They are very important in terms of helping the conducting of the neuronal impulse down the length of the neuron. These are Schwann cells that you see in here, which are very important glial cells that are crucial for protecting the axon and helping in the development of the myelin sheath. So again, the basic building block of our behavior is the neuron. And again, this is the receiving area, and down here, this is, once the neuronal impulse, which we will talk about, travels from the cell body down the axon to the axon ending. It's having very important effects in these axon endings that you see right here. So let's talk a little bit more about this. Let's classify cells in two basic ways. We can classify them in terms of whether they have afferent axons or efferent axons. And the diagram that you see here of two different nerve cells, afferent axons, like what you see right here, okay, those afferent axons are bringing information into the cell body, okay? So they're referred to as afferents. Efferent axons, on the other hand, means carrying information away from the cell body. So what you see right here is an efferent axon that's carrying information, this neuronal impulse, from the cell body down the axon and then to this adjacent nerve cell that you see here. So again, we can classify cells as being either afferent or efferent. So let's talk a little bit about the action potential, and hopefully this diagram is going to help you to understand how this information, this neuronal impulse, goes from the cell body down the axon and then ultimately to this axon terminal that you see here. When we examine the neuron, there's two very important changes that are going on that's responsible for the action potential. One is a change in terms of what we call the permeability of the membrane, and the other is a change in terms of the voltage, that is the electrical nature of the membrane. So what you see in this diagram right here and right here, let's just deal with this part that we see here, okay? And when the neuron is resting, that is, it's not firing, the voltage across the membrane is about minus 70 millivolts. So that's what you see right here. And here's our membrane of our axon that you see right here. And there's a lot of sodium on the outside and a lot of potassium that's on the inside. Okay, that's in the resting state. Now let's move to the second part of this. What we have right here now is this voltage now is changing. It's becoming less negative, more positive. And what you have inside the cell is you have sodium that is rapidly going inside the cell. Okay, so now what we have is a change that's occurring in terms of polarity. A lot of that change is occurring as a consequence of these charged ions, sodium and potassium. So again, you have a lot of sodium now that's going inside the cell. And then what happens is the neuron fires. When it fires, this is associated with a change that is occurring with now potassium rushing outside of the cell. And indeed, here's the end of the impulse that you see here. Again, you're going back now to more of a polarized state, a negative state. So indeed, there are electrical changes, there are biochemical changes that are occurring that are responsible for pushing this neuronal impulse down the axon to the terminal what's called the terminal button area that you see right here. Let's take another view of this. What is happening? Here's our axon, and here's our action potential, which is traveling from this direction down to this direction. Sodium channels are opening up, sodium ions are entering, and it's now changing that membrane potential, okay, which was mainly negative. Now it's being changed to being more positive. In the second stage, potassium channels are opening up, potassium ions leave, they're reaching that membrane potential, and then the ion, what's called the sodium-potassium pump now, these transporters now are transporting sodium and potassium back to their original location. So that's what's responsible for pushing now this electrical charge down the length of the axon. So the action potential, the firing of a neuron, depends upon two fundamental processes. One is a change in permeability of the cell membrane to sodium and potassium. The other is a change in the polarity, the charge of the neuron. The neuron, instead of being in this negative state, is becoming more positive. Then once it fires, it becomes more negative again and goes back to its resting potential. Here again is what is happening in terms of that change in polarity of the action potential. Here it is at rest. Let's say a stimulus is applied to a neuron. We'll talk a little bit about that as we go along. And once it gets up to what we call threshold, where firing can occur, the voltage is rising, right, up to about plus 30, right? And then the neuron fires, and then the voltage now goes down and ultimately returns to a resting potential. So again, this now represents the end of the action potential. So this change in polarity then is extremely important, as well as the change that occurs in terms of the permeability of the membrane. So electrical changes in the membrane, now let's associate them with what's happening in terms of sodium and potassium. Top part of this figure shows what's happening in terms of this electrical potential. Bottom part shows what's happening at the same time in terms of the rate of entry of sodium into the neuron and the rate of exit of potassium from the neuron. So again, let's take a look at this again. Minus 70 millivolts, right, gets up to threshold, it fires, right, goes up to about plus 30, plus 40, then comes down and eventually comes back to its original minus 70 millivolt resting state. Here's what is happening. These are the associated changes in terms of sodium and potassium. In the red, take a look. This almost completely mimics what is happening here. That is the entry of sodium into the neuron. Okay, now it goes down again after firing takes place. The rate of exit of potassium from the neuron, that's in the blue. This occurs a little bit later and then eventually goes down. So again, associate with what is happening here in terms of change of polarity, change of the electrical potential, with what is happening here in terms of the rate of entry of sodium and the rate of exit of potassium. Okay. Let's talk a little bit about this gap here that's called the synapse. Again, here's a neuron, and here's the terminal button area that you see here. Okay, and if we were here, it's resting upon an adjacent neuron. This is a blow-up right here of what this looks like. Here's the approaching nerve impulse, and there are these what are called vesicles. They're like little packages. And these red dots that you see here refer to as neurotransmitters. They are pushed down to this, what's called the presynaptic area, where those packages open up and release neurotransmitters out into this gap, out into what is called the synapse. Here's the postsynaptic membrane that you see here. Again, this is right here. And this is where the receptors are. They operate according to kind of like a lock and key arrangement. Certain neurotransmitters unlock certain neurotransmitters. So what happens is that those neurotransmitters now lock into those receptors, and then they cause this adjacent neuron to fire. So indeed, this is the chain of events that occurs at the synapse. Take a look now at another view of this postsynaptic area that you see. This is the postsynaptic area. This is the presynaptic area. This is where those molecules of neurotransmitters, these chemical substances, are secreted. And again, what they're going to do is they're eventually going to come down into these receptor sites, and they're going to unlock those receptor sites. Now, what can happen is you're either going to get excitation here in this postsynaptic membrane, or you're going to get inhibition. One of the two things can occur. But again, this is something that is referred to as postsynaptic firing. And again, this is all related to the occupation of these receptors that you see here by these neurotransmitters, again in a lock and key arrangement. There's also a phenomenon that is referred to as reuptake, which you should have a little bit of familiarity with. Take a look here once again. We have activation that is occurring of this membrane, this postsynaptic membrane, because these neurotransmitters have been released out into this gap where they're taken up by these receptors. But what can also occur is a phenomenon called reuptake, in which those molecules can actually go back, those neurotransmitters can go back, feedback upon the presynaptic area. They are transported back by what are called transporter proteins, and they in fact can modify the production as well as the release of those neurotransmitters in the future. So this is something that is referred to as reuptake. Again, you have these neurotransmitters that are being taken back up into this presynaptic area that you see right here. Okay. So again, very important. to understand both of these processes because both are involved in behavior. We're going to continue on with an assessment of this in our next lecture and understand a little bit more about those neurotransmitters and their various actions they have in terms of our behavior.